Welcome back to Environmental Science. This is Chapter 2. We're looking at Lesson 8. In this chapter, we're going to be looking at tools for science. Science in general, as well as science in the environmental realm. So let's talk about scientific methods. The word science comes from a Latin word called scire, which means to know. In science, we have a lot of interesting facts, ideas about how nature works, but science is not just something you know, it is also something that you do. In this chapter, we're going to learn how science is done and examine some of the tools that scientists use in order to do their work. One of the main tools that they use is something called the scientific method, or sometimes it's called the experimental method. You've probably heard the phrase something like, today scientists discovered, and then fill in the blank. How do scientists make these discoveries? Scientists make most of their discoveries using the experimental method or the scientific method. This method consists of a series of steps, and although those steps don't go in order in every situation, most of them are there. These series of steps help scientists to identify and answer questions. The first step is usually some sort of observation. We see something that's out of place, something that didn't happen before. We make some sort of observation that causes us to question or to wonder. Observation is simply a piece of information that we gather using our senses, such as our sight, our hearing, our touch, even our sense of smell. So we use our senses to make these observations, and then we extend our senses by using equipment that may pick up things that we are not able to perceive. We might use an amplifier to amplify sound. We may use a microphone to pick up very small sounds. We use seismographs in geology when we're studying earthquakes. So there's lots of different instruments that we can use that help us to not only pick up and extend our senses, but in some cases they allow us to do measurements more accurately. We use rulers, so we don't have to say, well, it was a long distance. Instead, we can give a number and a unit, such as it was 12 kilometers or it was 3 feet. So we have a number indicating how many units there are, but then we also indicate what kind of units we're using. 3 feet is dramatically different from 3 miles. We can use satellites to pick up information about our atmosphere and things that are going on the Earth. Rulers provide our eyes with a standard way to compare the lengths of different objects. Scientists can observe even something like the tail length of a wolf to help them understand how the wolves are living and what's going on in their environment. So they may tranquilize a wolf and then use a tape measure to measure the length of the tail. Observations can be made in a lot of different forms, including descriptions. We can have drawings. In today's world, there's a lot of photographs. And then we can also take those measurements. As you go about your daily lives, you might see something that wasn't there before. And you might notice something, a problem of some sort. And what you need to take steps in order to correct that type of problem. Well, in order to correct the problem, we really have to understand the problem. So the first step in the scientific method or the experimental method is observation. And with that observation, we might gather some basic information. Once we have some of that basic information, we're likely to form some sort of hypothesis. So observations give us answers to certain questions, but observations almost always lead us to more questions than what we do answers. Okay, well, this is what happened, but why did it happen? Would it always happen that way? Does it happen that quickly? Can we speed it up? Can we slow it down? So to answer specific questions, what scientists may do is they may form what is called a hypothesis. A hypothesis is a testable idea or some sort of explanation that leads to scientific investigation. A hypothesis is more than just the educated guess that you may have learned in elementary school, maybe even middle school. A good hypothesis follows what the information that we already know. It can be tested, And many times it tries to make some sort of prediction. So if we can put a number to it, then that allows us to quantify how close our hypothesis is to meeting all the facts and the data that we're going to gather as we do experiments. So let's just say I wanted to find out, do car seats save lives? Now, there's lots of information out there already, but let's just take this as a for instance. Let's say seatbelts were brand new, haven't really been tested and find the data. So what kind of things could I do to test seatbelts? Well, I would take perhaps different vehicles of some sort. It doesn't necessarily have to be a car. It could be just like some sort of buggy or train that we can use to test. I can strap like a car seat in or some other figure, even a sack of potatoes in, and we can strap it in, and then we can ram that going at various speeds and seeing what happens to that sack of potatoes or the sack of flour or whatever it is that we have in there. So what we do is we form a hypothesis. Our hypothesis is that seat belts would decrease the amount of damage done in an accident. And so that's our hypothesis. And now what we need to do is we need to come up with something that can quantify our hypothesis. That helps us to determine, is our hypothesis close or are we far off? And so maybe I make predictions that potatoes will not be as bruised. Right? Maybe I want to quantify that even more, that they'll be bruised half as much. If we were testing water quality in a river, let's say that somebody noticed that the mussels or some type of fish in the stream had issues, their numbers seem to be going down. Well, we might make some predictions as to the water quality. Maybe there's too much phosphates in the water, and maybe that chemical is decreasing. Well, if that's true, then I might ask, well, where did that chemical come from? Is it from fertilizers from some of the farms, or maybe people are fertilizing their lawns and it's washing into the stream? So armed with these observations that we've made, we can follow to the hypothesis phase. We can make our hypothesis, we can make that prediction. A prediction is simply some logical statement about what's going to happen if the hypothesis is correct. So in our example of the mussels or the fish, we might make predictions like the mussels will die if we expose them to high levels of phosphate. It is important that we understand our hypothesis, that our hypothesis deals with the high levels of phosphate that are killing mussels. We may be able to test it to where we can either prove or disprove. Really what we're looking more for is not proving our hypothesis correct, but rather making more and more evidence that we cannot disprove our hypothesis. A good scientist is going to constantly try to prove themselves wrong, because if we can't prove ourselves wrong, then that means we are probably correct. So if the students, in our example, if we were to try to successfully raise mussels in water that had high phosphate levels, then that would prove our hypothesis incorrect, because our hypothesis states that mussels aren't going to survive in high phosphate water. So if we're able to raise mussels in an environment that has high amounts of phosphate, then we've proven our hypothesis wrong. That's actually a good thing. That's one fewer things that we have to look at as possible solution or cause, and then we can go on to the next. So every time a hypothesis is disproved, our possible explanations for an observation or a reason for an observation is reduced. By eliminating possible explanations, a scientist can zero in on what the best explanation is for a certain event occurring. And that increases our confidence in our final answers. So once we come up with a hypothesis that's testable, the next step is to actually test it, experimenting. The questions that arise from observations often cannot be answered making more observations in and of themselves. In the situation, a scientist usually performs one or more experiments, which is a procedure designed to test a hypothesis under controlled conditions. Unfortunately, with environmental science, there's a lot of things that we cannot control. If we're doing an experiment outside, we cannot have complete control over the weather. We cannot have complete control over wind factors, over insects. There's a lot of things. So what we do is we try to bring it into a laboratory situation where we can control how much rainfall there is, how much wind there is, the insect amount mostly. So experiments should be designed to try to pinpoint the cause and effect relationships, specifically that involved with your hypothesis. For this reason, good experiments have two essential characteristics. A single variable is tested and a control is used. So a variable, what is a variable? Well, if you think about mathematics, what is a variable in mathematics? It's a letter or some symbol that changes based on what's going on. So in a scientific sense, a variable is a factor that we're interested in, which in our example was the phosphate levels of water, that we can change purposely. So to test for one variable, scientists usually study two groups or two situations at a time. There may be more than two. We might be testing 5 different phosphate levels, but we generally have at least 2. The variable being studied is the one that's the only difference among the different groups. So in our example of the phosphate levels with the mussels, we may have 5 tanks. In one tank, my phosphate level is 0, and I put 10 mussels in that tank. In the next tank, maybe I increase my phosphate level to a level 1, and I put 10 mussels, the same type of mussel, in that tank. Hopefully, roughly the same size, everything about them, as much as possible, being the same. In the next tank, maybe I do a level 2 phosphate, and I put 10 mussels of the same type and same size in that tank. So on and so forth with all the different tanks. And we test that one variable, and we look and compare all those different variables. So the variable being tested is the only difference, as much as possible, between or among all the groups. The group that receives the experimental treatment, or the groups that have the experimental treatments in our scenario where we have several different groups, that's called the experimental group or groups. In our example, the experimental group would be the different phosphate levels. The control group, which is the one that does not have the variable being tested, would be the tank that does not have any phosphate in it. That's our control group. That way, as we go through the experiment, we can look at the different sizes. What's the average size in tank 1, tank 2, tank 3, tank 4? How many of them are still alive? How many of them have died? And we can use that information to do a comparison. So the control group is what we're using as a comparison. Once we have done the experiment, obviously we are gaining data. So keeping careful and accurate records is extremely important in science. A scientist cannot rely on experimental results that aren't based on good observations and complete information. The information that scientists gather during an experiment is called data. It's usually in a numeric form as much as possible because numbers don't change. If I say something is blue, well, is it a dark blue? Is it a light blue? Is it kind of a bluish green? Is it an aqua? I mean, what kind of blue is it? But if we can put a number to it, then that really, really helps. If we can measure the mussels size, if we can count how many are there, if we can measure the amount of phosphate in the tanks, those are data. They're individual numeric forms of information. Once we have that data, we can organize it into tables. We might be doing graphics of some sort to see how the growth of the mussels took place throughout time. So any of those types of illustrations helps the scientists to analyze that data that they gathered and might be able to explain the data more clearly as they try to explain it to others. So the scientist is analyzing data on perhaps pesticides in food. Here we might be analyzing the data that we got from our different tanks as we were measuring the phosphate levels and our mussels. Graphs are often used by scientists to help display that relationship to others, especially if they're giving it to non-scientific realm people. Graphs are especially useful for illustrating conclusions that are drawn from the experiment. One common type of graph is called the bar graph. Bar graphs are used for comparing data of several different things in one graph, or several different groups in one graph. So if we looked at the concentration levels of these different groups or different tanks or different sites of phosphate, we might be able to take a look at their information in a graphical manner. The one that has the higher amount of phosphates has a taller bar, and we try to keep it to where they are proportional among all of them. So once we've done our experiment, we've gathered our data, we've organized that data, now it's time to draw our conclusions, at least our initial conclusions. So scientists determine the results of their experiment by analyzing the data and comparing the outcome with their experiment with their predictions. How did my hypothesis line up with my actual data that I got when I collected from my experiment? Ideally, this comparison provides scientists with obvious conclusions. Either it fell in line with my hypothesis or it totally destroyed my hypothesis. Either one is a benefit, but often the conclusion is not that obvious. For example, in our muscle experiment, if 3 muscles died in our control group and 5 died in our experimental tanks, well, can we be certain that it is the phosphate that's causing them to die? Those numbers are still pretty close. Perhaps I just happened to pick some more healthy muscles that went in the control group, and maybe some of the weaker muscles went in the other groups. I didn't do it purposely, but it could happen. So scientists often use mathematical tools to help them to determine whether the differences are meaningful or not, or just a coincidence. That is more of a math field. We're not going to talk about that. It's often referred to as the chi-squared value. Mathematics, you might be able to do that when you go into statistics. For us, we just need to recognize that we're comparing the groups. We're probably going to do some sort of math in order to determine, is it significant or is it not significant? Once we've drawn those conclusions, that's not where we stop. We got to keep on going. What I'm going to do next is I'm going to repeat my experiments. Although the results from a single experiment may seem very, very conclusive, very solid, very good, lines up exactly with what we thought or lines up exactly against what we thought, we still have to be careful. Scientists look for large amounts of supporting evidence before they accept a hypothesis. If you're going to publish your information, you want to make sure that you've done a lot of experimentation. You don't want to just do it once. You maybe want to do it 100 times. And then, if you get the same results time after time after time, then you feel, every time you do one of those experiments and you get the same results, you feel more and more confident about your answers. So the more sure scientists become about the reliability of their conclusions, the more apt they are to go to the next step of the scientific method, which is to communicate those results to other scientists. Scientists are very skeptical. They don't believe much. And so when they read something that doesn't seem quite right to them, their initial thought is, I'm going to do the same experiment and see if I get the same results where I am. So scientists publish their results to share what they have learned with other scientists. When scientists think their results are important, they usually publish their findings in some sort of scientific article or journal. A scientific journal includes the question that the science explored, the reasons why the question is important, at least to that particular scientist, maybe some background information, and then a precise description of how the experiment was done and what kind of data they collected and what their data resulted in. Now, they may not give all the individual pieces of data, but they usually do it in a form of an average of some sort. And then the scientist's interpretation of what that data means. Now, here's the problem, one of the main problems with the scientific method. Not everything can be experimented, which brings us to the correlation method. Whenever possible, scientists study questions by using experiments, but many questions cannot be studied experimentally. The question, for instance, what is the Earth's climate like 60 million years ago, cannot be studied by performing experiments because we don't have experiments from 60 million years ago for several reasons. Humans weren't even alive back then, and the earth wasn't even around back then. But even talking about, well, what was the environment like prior to Noah's flood? There's no experiment that we can do to test that. Here's another example of something that can't be tested. Does smoking cause lung cancer in humans? We cannot experimentally study that question because in order to study it scientifically, we would have to get a random group of people, break that random group of people into two different groups, say, none of you in this group can smoke, and all of you in this group must smoke. We would have to force smoking on individuals. That would be unethical. So, when using experiments to answer these kinds of questions where the question is impossible or unethical in order to try to experiment with, we try to make predictions based off of correlations. A correlation is an association between two or more events. For example, scientists may know that the relative width of a ring on a tree is a good indicator of the amount of rainfall and sunlight that tree received during a given year. Trees that produced thicker rings or wider rings typically grew during rainy years, and the narrower rings tend to happen during the drier years. So scientists have used this knowledge to investigate why the first European settlers in Roanoke Island, Virginia, often called the Lost Colony, disappeared, and why most of the first settlers in Jamestown, Virginia, also died. Now, if we look at the tree rings in those areas for the years, and we backtrack back to the years that Roanoke and Jamestown were founded, we find some interesting things. Two of the worst droughts the East Coast had experienced in centuries happened during those years that those two colonies were founded. So therefore, it leads us to conclude that perhaps the reason that the lost colony is lost and a lot of settlers in Jamestown died was due to famine. Famine because it was very difficult to grow food because of the lack of rainfall. All right, now although correlation studies are useful, correlations do not necessarily prove cause and effect relationships between the two variables. For example, the correlation between increasing phosphate levels and a declining muscle population does not necessarily prove that the phosphates harm the muscles. Scientists become more and more sure about their conclusions if they find the same correlation in different places as they eliminate other possible explanations as well. So it takes more, right? We see that smoking is linked to lung cancer, an increased probability of lung cancer. Now, we had to use correlations in order to do that because we can't force people to smoke. But what we can do is we can find people who have lung cancer and ask them, did you smoke? How much have you smoked? And we can get some information from them. And we can look at non-smokers and see their rates of lung cancer. And we see there's a distinct correlation, perhaps, between the two groups, people who smoke and people who don't smoke. People who smoke have a much higher rate of getting lung cancer than those non-smokers. However, we can't definitively say that it causes lung cancer because perhaps there's something innate in their DNA that causes that person to want to smoke. And perhaps that same DNA strand that causes that individual the desire to smoke more than the average person would also be a DNA strand that would increase their likelihood of getting lung cancer. So we don't know for certain, but there is some sort of link between the two. So as we look at scientific method, we have lots of different pieces to it. We start off with observing, which is gathering pieces of information through our senses. Those senses can be aided with certain tools, such as measuring devices. Then we move on to a hypothesis, which is more than just an educated guess. It is a possible explanation that leads to a scientific investigation that is testable, that we can measure. Once we have that, then we can start making predictions based off of our hypothesis. And then that leads us to making and designing an experiment. An experiment is a procedure designed to test that hypothesis under controlled conditions. We have two types of groups in our experiment. We have a test group, what is known as the experimental group, and then we have a control group. The experimental group gets differences in our variable. Whatever we're testing, that's what we're going to change in the experimental group. The control group, as much as possible, is going to be kept constant. Once we are performing that experiment, we want to make sure we gather as much data as possible. That data could be in the forms of pictures, but most often it is in the form of numbers. Gather data as the experiment goes on. Data is simply information in a numerical form. Once we have that data, we try to draw conclusions. Helping us draw conclusions, we might do some graphs, we might do some other things that will help us organize and analyze the data that we collected. So we're going to draw our conclusion. Once we think we have our conclusion, we go back and we re-experiment, making sure that we have as much control as possible. So we keep on experimenting. Every time we experiment, we get the same conclusion. We feel more confident in our conclusion. Once we feel confident enough, we can start communicating those results to other scientists so that they can also perform the experiment and hopefully increase the confidence that we have in our own results. Now, everything, however, is experimental, and so sometimes we have to use correlations. If we cannot experiment with something or it is unethical to experiment, we need to use more of a correlation design to our quote-unquote experiment.